In this video, I wanted to talk a little bit about the ingredients that are found in the mRNA vaccines for COVID-19. And so this is a list of the ingredients from both the Moderna and the Pfizer vaccines. And as you can see, they are similar in a lot of ways. So they both have the messenger mRNA. They both, well, this one has this tromethamine and tromethamine hydrochloride, acetic acid, sodium acetate. And this has the potassium chloride, monobasic potassium phosphate, sodium chloride, dibasic sodium phosphate dihydrate. And, they're chemical names, so it's always a little bit, I think people can be a little put off by chemical names, but essentially what these chemicals are doing is just making it a salt solution. I mean, that's what the sodium chloride is. That's just table salt. Potassium chloride is just I mean, that's another salt that everybody has in their bodies. Then this monobasic potassium phosphate and dibasic sodium phosphate are what are called buffers. And so that's just to control the pH of the solution. Then sucrose here, that is table sugar, and that acts as a cryoprotectant. So these vaccines are stored at very low temperatures, and when you decrease the temperature like that, it causes crystals to form, and that can damage the lipid nanoparticles that these vaccines are composed of. And so sucrose helps prevent that crystal formation so that it does not damage the lipid nanoparticles. So then this mRNA is sort of the business end of this. And so mRNA has a structure that's sort of like this. So this is actually DNA. If this were RNA, then this would actually be a hydroxyl group here. So DNA is deoxy because it gets rid of those oxygens there, but in RNA it's not deoxy, it's just ribonucleic acid, so it does have those oxygens there. But sort of, well, in these vaccines, these bases are going to be somewhat modified. So usually that's sort of, putting maybe like a methyl group onto this or things like that in order to help protect the mRNA from being broken down inside the human cells. But the mRNA that these actually have is an mRNA that codes for a part of the spike protein. And so then the spike protein can be presented on the surface of the cell, and that acts as what's called an epitope. So an epitope, which then immune cells can actually sort of see that and say, oh, this thing is something that shouldn't be here, so I'll start making antibodies against that. And that in this case would be the spike protein of the SARS-CoV. And I've talked about how this works in a bit more detail in other videos. But the one thing to sort of notice here is this phosphate backbone, and these are negatively charged, and negative charges are called anions, and that will be important to remember here in a little bit. Because the other thing that I wanted to talk about, which is probably even a little bit more scary looking, are these lipids here. And those are the things that make up the lipid nanoparticle. And so we can see that the Moderna also uses lipids. So they use a little bit different of lipids than the Pfizer one, but they are behaving, they're doing sort of the same job. And so I am just going to focus on the Pfizer BioNTech version of this. And so there are three different kinds of lipids, and I have the structures of them right here. So there's this one, and they have these big, long, complicated names. But in organic chemistry, these big, long, complicated names are essentially just saying everything that is inside this molecule. So it has this 4-hydroxybutyl. So the 4-hydroxybutyl is going to be 1, 2, 3, 4. So I was trying to draw that with my mouse, but it's 1, 2, 3, 4 carbons, then this hydroxyl group, and a 4-carbon chain is a butyl group, or butane. Butane, you might have heard, is in lighters, and that's just a 4-carbon chain. And then, I'm not going to go through naming conventions for organic molecules, but the point I'm making is that all this, sort of complicated stuff is just sort of supposed to tell a chemist what this molecule actually is. A person who knows what all this stuff means would actually be able to draw the chemical structure just based on the name of it. But this also is oftentimes just called ALC00315. So ALC0315. Then we have this other one here, which so I guess the thing to sort of notice on this is this red part here, which means, and it has this N here because it can be N number of these things in parentheses repeating long. And so that is a polyethylene glycol coming off of it, and 2000 because that is sort of the average length of this polyethylene glycol part here. Then it has the N-ditetradecylacetamide. So it has the NN because they're both coming off of the N right there. And then ditetra, so ditetra decylacetamide, which is just talking about these two chains here. Then we also have this lipid here, which sort of the business end of this is that it has this group right here, which, as you can see, has a negative part and a positive part. So that is what's called a Zwitter ion, when it has two full charges that are opposite charges of each other. So I copied this out of a paper that talks about these lipid nanoparticles, and it sort of explains what these lipids are doing. So this is from a paper that actually predates COVID, and it was actually looking at using lipid nanoparticles for cancer treatments. But we can see we have a lot of the same things here. So this is a typical lipid nanoparticle formulation. Is composed of pH responsive lipids, or cationic lipids, so cationic being positively charged lipids bearing tertiary or quaternary amines to encapsulate the polyanionic. Remember, anionic was those negative charges on the phosphates on the mRNA, so the polyanionic mRNA. So it's polyanionic because it's anions, but it also has, multiple of them, so it's polyanionic. And so the tertiary or quaternary amine is what we can see right here. So this is a tertiary amine right there, and that is the tertiary amine that it's talking about. And so that amine can become So a hydrogen can actually bond to that nitrogen there based on whether it's in the right pH. So at low pH, you can actually get a hydrogen coming off here, making it positively charged. And so that positive charge will become attracted to the negative charges on the mRNA. And so it can actually sort of form an ionic bond with that negative charge on the mRNA. And so that's what that is doing. So then it says the neutral helper lipids, such as Zwitter ionic lipids. Then it lists these two different kinds. So the 1,2 diol SN-glycerol 3-phosphoethanolamine or dope, and then DSPC. And so that is what we have right here. So this is 1,2 disteroyl SN-glycerol 3-phosphocholine. So this is Zwitter ionic part here, the positive charged nitrogen, and the negative charged phosphate is the choline part, and that is the Zwitter ionic part, and or sterolipid, i.e. cholesterol. And so we can actually, I don't have a structure of it here, but this does say that cholesterol is part of the lipid ingredients in there. To stabilize the lipid bilayer of the lipid nanoparticle and to enhance mRNA delivery efficiency, and a polyethylene glycol lipid to improve the colloidal stability in biological environments by reducing aspecific absorption of plasma proteins and forming a hydration layer over the nanoparticles. And so this is that polyethylene glycol right here. And so it's saying that that is to improve the colloidal stability. And so a colloidal solution is, well, it's what these lipid nanoparticles would be. So we have here our solution, and they would actually be these sort of fully formed lipid nanoparticles. They are not being dissolved into the solution. They are fully formed in that in these lipid nanoparticles. In fact, I believe milk is actually a colloidal solution, and that's why, it's sort of milky white. But anyway, I'm not going to get into that. So it helps stabilize these colloidal forms, so these lipid nanoparticles don't break apart, and in particular in biological environments, by reducing the absorption of plasma proteins. So these lipid nanoparticles won't absorb proteins, and forming a hydration layer over the layer of nanoparticles. So a hydration layer is essentially, we have our nanoparticle here, and a hydration layer is just water, so H2O sort of around our lipid nanoparticle here, and so it's helping form that hydration layer. And so that's what those three different lipids are doing in the vaccine. And so this is a schematic of a lipid nanoparticle here. And so these little sort of football shapes are the head of the lipid nanoparticles. So the head on this one would be this part right here. The head on this one would be this part right here. And this Zwitter ionic part over here would be the head on that one. And so you can actually see here they have these red plus ones here, which are supposed to be this head right here, I believe. Yeah, because those ones also form these little shells around these little red spaghetti noodle things are the RNA inside of it. And so those the positive charge on that nitrogen is associated with the negative charge on the mRNA. Then you can see these blue ones with this tail like this. That tail is this part right here. It's that polyethylene glycol part coming off there. And you can see these tails coming off here are supposed to be helping stabilize this in the lipid nanoparticle form. Then these little yellow things here are the cholesterol. And so when these lipid nanoparticles come up to a cell, they actually enter the cell through endocytosis. And a paper I was looking at was saying that what they want is for these lipid nanoparticles to be able to escape the endosome here, escape the vesicle out into the cytoplasm before going through this process of having the endosome change into this lysosome, because a lysosome is essentially the part of the cell that breaks things down. It's like the cell's digestive system in a way. And so you want these lipid nanoparticles to escape from these endosomes early on before it gets to that lysosome stage. And the endosomes evolve by having different proteins on the surfaces of them and having different phospholipid bilayer composition. And I'm not going to go too deeply into that, but this is the way that that these lipid nanoparticles are brought into the cell. And so I have that paper right here. And yeah, let's see, are engulfed into the cell. They follow the conventional endocytic route. However, increasing evidence indicates the endosomal escape mechanism of lipid nanoparticles is much more complex. And anyway, this will be linked to in the description down below if you want to read more about how these sort of enter the cell. But the idea then is that the lipid, or that the mRNA actually escapes out of the early endosome, and then the mRNA is in these cell plasma, the cytosol, and can then be translated. The mRNA is then translated there by ribosomes into a part of that spike protein, which the cell will then sort of recognize as not something that should be there. And the cells will actually then sort of present that on the surface of the cell so that immune cells can sort of latch onto it and recognize and say, this shouldn't be here. Let's start making an immune response against this. Let's start making antibodies against this spike protein, and and then your immune system will remember that spike protein, and so it can actually mount an immune response much quicker in the future if you are exposed to something that has that spike protein, in this case COVID-19. But anyway, I hope you found this video somewhat illuminating as far as these mRNA vaccines go. I'm not trying to say one way or the other, whether these are dangerous or not, because it is a pretty new technology, but from what the data shows is that these are pretty safe to use. And in a lot of ways, one of the things that a lot of papers that talk about this actually sort of harp on is how these are safer than the previous, the attenuated viral particle style of vaccine, which has all these adjuvants and stuff in it that, can, they're not dangerous, but they can cause, sort of more adverse reactions and things like that. And so, these are actually potentially much safer than the old style of vaccines and will hopefully cause less adverse effects. So, like I said, these lipids, these big long lipid names, are probably something that people will be sort of wary of when they see them because it's like, wow, look at these crazy, complicated chemicals that I can't even pronounce. That must be something really weird and alien and foreign and stuff. But I mean, they're just these sort of lipid particles here. I mean, they are strange lipid particles. They're not like the fats that you usually get from your diet, but they're not something that's terribly weird or anything like that. And then the other thing that uh, people might be wary about is the mRNA itself, which you know it seems it always seems a little bit weird with vaccines that you're sort of exposing yourself to something that's potentially dangerous, but this is only exposing you to a very small part of the COVID-19 genome. So, the COVID-19 genome is this really long RNA. Then there is a section of it that is the spike protein, and then the vaccine is only giving even just a small section of that spike protein. It's not even giving the full spike protein, and so it's not something that can infect you. I mean, the effects people have, with some people like feel a little bit sick after taking it. That's actually your immune response. So when you're sick, a lot of these symptoms that you experience are actually the result of your own immune response. And so this is not actually making you sick. And the other nice thing about mRNA is there is no chance of it actually, messing with your DNA genome. So if we were using DNA instead of RNA, there would be a chance that it could actually sort of incorporate into your own genome and cause problems that way. But mRNA cannot do that, and so that is another thing that makes it safer. And like I said, these four things here are just essentially salts and pH buffers. Those and things like potassium chloride, sodium phosphate. Those are all things that are found in human blood already anyway. And so, dibasic and monobasic is just telling you essentially how many potassiums or sodiums are on the phosphate, because phosphate is a molecule that looks like this. So it's a P, well, actually, that's just a single bond there to an O. So it's a phosphate bound to 4 oxygens. These 3 oxygens with the single bond are all negatively charged. And so if you have a monobasic potassium, you would have a potassium right here, and then you would have hydrogens on here. Whereas if you had dibasic potassium, it would be like this: 2 potassiums and then a hydrogen. They call it dibasic because in solution, those potassiums will dissociate from this and actually become these negatively charged oxygens. This would probably still have the hydrogen on it. And these oxygens are bases. They're alkaline, so that's why it's called dibasic. And like I said, the sucrose is a cryoprotectant. It helps prevent the formation of crystals that could damage these lipid nanoparticles. So you don't want water crystals forming that could kind of jam their way in there and start breaking things apart. And so sucrose helps that for storing it at low temperatures. But anyway, I feel like I'm sort of rambling on now. I hope you found this video helpful, and I will see you in another video.